Calvin cycle of photosynthesis. Photosynthesis. The overall process converts carbon dioxide and water into glucose and oxygen. It consists of two main stages, the light reactions and the Calvin cycle, where photosynthesis occurs. Photosynthesis takes place in the chloroplasts of mesophyll cells within plant leaves. Chloroplasts are specialized organelles that capture light energy and fix carbon dioxide for glucose production. Calvin cycle. This cycle occurs in the stroma of the chloroplast and was discovered in the late 1940s by Melvin Calvin, Andrew Benson, and James Bassel. Steps of the Calvin cycle. The cycle consists of three main stages: carbon fixation, reduction, and regeneration of ribulose 1,5-bisphosphate. Carbon fixation. Carbon dioxide is fixed to ribulose 1,5-bisphosphate, a 5-carbon compound, forming an unstable 6-carbon intermediate. This splits into two molecules of 3-phosphoglycerate, each with 3 carbon atoms. The enzyme ribulose 1,5-bisphosphate carboxylase oxygenase, Ribisco, catalyzes this reaction. Ribisco can also bind oxygen, leading to photorespiration. Reduction. Each 3-phosphoglycerate molecule is converted into glyceraldehyde 3-phosphate, a 3-carbon compound. This process consumes adenosine triphosphate and nicotinamide adenine dinucleotide phosphate. The reactions are catalyzed by 3-phosphoglycerate kinase and an ADP-dependent glyceraldehyde 3-phosphate dehydrogenase. Some glyceraldehyde 3-phosphate molecules exit the cycle to form glucose. Regeneration of RUBP. Glyceraldehyde 3-phosphate is rearranged to regenerate ribulose 1,5-bisphosphate, the 5-carbon carbon dioxide acceptor. This step requires ADP and enzymes including aldolase, transcetylase, fructose 1,6-bisphosphatase, and phosphoribulokinase. As a summary, in carbon fixation, RUBP reacts with carbon dioxide to form two molecules of 3-PGA. In reduction, 3-PGA is converted into G3P using ADP and NADPH. Finally, part of glyceraldehyde 3-phosphate is recycled to regenerate RUBP as completing the cycle. Photorespiration. Under high oxygen concentration, Ribisco oxygenates ribulose 1,5-bisphosphate instead of fixing carbon dioxide. This produces one molecule of 3-phosphoglycerate, a 3-carbon compound, and one molecule of 2-phosphoglycolate, a 2-carbon compound. Recycling 2-phosphoglycolate consumes adenosine triphosphate without producing glucose, reducing photosynthetic efficiency by up to 50%. This process requires the chloroplast, peroxisome, and mitochondria. C3 to C4 and C them. These adaptations help plants concentrate carbon dioxide and reduce photorespiration. C4 pathway. In C4 plants, carbon dioxide is initially fixed in mesophyll cells by phosphoenol pyruvate carboxylase, forming oxaloacetate, a 4-carbon compound. This enzyme only fixes carbon dioxide and does not react with oxygen. Oxaloacetate is transported to bundle sheath cells, where carbon dioxide is released and enters the Calvin cycle, allowing efficient carbon fixation without oxygen interference. CM plants. In these plants, carbon dioxide is fixed at night by phosphoenol pyruvate carboxylase, forming 4-carbon organic acids stored in vacuoles. During the day, carbon dioxide is released from these acids and enters the Calvin cycle, minimizing photorespiration. Real-world applications. First, crop improvement. Manipulating Calvin cycle enzymes can increase photosynthetic efficiency and crop yield. Second, climate adaptation. Understanding photorespiration and C4, CM pathways allows the development of crops that withstand heat and drought. Third, bioenergy production. Optimizing carbon fixation in plants and algae supports biofuel development. Life depends on energy flowing through the world, from the sun to plants, and from plants to all living beings. The Calvin cycle keeps this energy moving, connecting all forms of life. Truly, it is the heartbeat of life. The Calvin cycle is life's energy source.